So this chapter will hover around these points. We have to study de novo synthesis and salvage pathway of biosynthesis of nucleic acid, purine, pyrimidine biosynthesis, and their breakdown process which produces uric acid, the associated clinical condition called gout, Leschnian syndrome, etc. Next. Now, what are these purines and pyrimidines? They are called nitrogenous base. These bases are found in nucleic acids, correct? They are called nitrogenous bases. Now, these nitrogenous bases has got two rings. Purine ring is made up of two rings. One is pyrimidine ring, another is imidazole ring. Purine ring is formed by fusion of two rings. Huh? Phone. Why? Why? 10 বছরের বাচ্চা খাতা খোল। So purine and pyrimidine, these two rings, purine ring is formed by fusion of one imidazole ring and one pyrimidine ring. Okay. How are they numbered? See the numbering. Pyrimidine ring is clockwise numbered, starting from the bottom nitrogen. These are all heterocyclic rings. So we start from clockwise 1, 2, 3, 4, 5, then 6. 1 and 3 are nitrogen. Purine ring numbering is a little bit different. We start with this meta nitrogen 1, 2, 3 means anti-clockwise, first this way, then this way. Anti-clockwise, first 1, 2, 3, 4, 5, 6, then 7, 8, 9. First anti-clockwise, then clockwise. This is how purine rings are numbered. You should remember the numbering of purine and pyrimidine ring. This will be useful. So this is basic purine ring, this is basic pyrimidine ring. Now these are the structures of commonly found purine and pyrimidine bases in DNA and RNA, correct? So DNA contains adenine and guanine, same for RNA, but RNA contains two pyrimidine rings which are different from that of the DNA, correct? RNA contains uracil and cytosine, DNA contains thymidine and cytosine. Uracil is missing in DNA and thymidine is missing in RNA. Are there any exceptions? Are there any exceptions? Yes, tRNA which contains thymidine, right, as unusual basis. So see the structures of adenine. The adenine is simple. What is the number of this carbon? Yes, this number, this carbon. Just now I told you to write down the numbering. Tell me by seeing that. What is adenosine? 6 amino purine. Guanosine, 1, 2, 3, 4, 5, 6 amino. Guanosine, this is 2 and 6, 2 amino, 6 oxo purine, right? Cytosine, this is guanosine, right? This is oxygen, this is NH2 group, if you just reverse it, it becomes cytosine in pyrimidine. Just reverse it, it becomes cytosine in pyrimidine. But this imidazole ring is missing. Then thymidine, same as uracil, is 1, 2, 3, 4, 2,4 dioxo pyrimidine. So same as uracil, except one methyl group is added at the 5th position. It becomes thymidine. So thymine can be formed from uracil by methylation, or if you remove the methyl group from thymine, it will become uracil. That is why there are some mutating agents which are demethylating agents, which are called demethylating agents. What they do? They demethylate thymine to form uracil. Now thymine is found in DNA, so when you remove methyl group, it becomes uracil. Now finding uracil in DNA is sign of mutation. So introduction of mutation can take place by demethylating agents. The agent compounds which will demethylate thymidine will create uracil in the DNA and which will lead to mutation, correct? So the compounds which demethylate are carcinogenic, at least mutagenic if not carcinogenic. Sugars, the nucleotide contains sugar plus nitrogen base plus phosphate. Usually the sugar is ribose sugar. Next, there are two types of sugar. DNA contains deoxyribose sugar. The 2' oxygen is missing in deoxy form, and the ribose sugar, it contains all the oxygen in place. Next. Okay, fine. Back here. Now, before going to the next structure, let us discuss. Very often we say that DNA is more stable compared to RNA. What makes DNA more? I gave you some hint. DNA contains deoxyribose sugar. So deoxyribose sugar is actually makes the sugar inert to other reacting substances. So other compounds cannot attack nucleophilically to the oxygen because there is no oxygen in 2' position, correct? So that's why presence of deoxyribose instead of ribose in DNA makes it more inert. Next. What is this? The structure of nucleoside. Adenine purine base attached with one ribose sugar, deoxyribose sugar or ribose sugar. Now this is ribose sugar. Which one is, this is C1, 2, 3, 4, 5, 6, 5, correct? If the 2' position is OH, then it will become deoxyribose. Deoxyribose plus adenine. So it will be deoxyadenosine. So when we say ATP, that is made up of ribose sugar. When we say deoxy ATP, deoxyadenosine triphosphate, then that is no more ATP. That is used for synthesis of DNA. ATP is not used for synthesis of DNA. Next. We will see why this difference. Again, we will see later. Structure of AMP and ATP. See, this is one phosphate. Then to that, again, if you add one phosphate, it will become ADP or diphosphate. One more you add, it will become ATP or adenosine triphosphate. If the sugar is deoxyribose, then just add one deoxy prefix. Deoxyadenosine triphosphate, deoxyadenosine monophosphate, deoxyadenosine diphosphate. Next. So we have got different names for different nucleotides. Next देखिए, नाम को देखिए, पढ़े जा. Here the names of nucleosides and nucleotides have been given. See, go through, read. Reading for once in a while, reading is also a very good habit. Right? So, base is adenine, nucleoside is, adenosine, nucleoside is adenosine, and nucleotide is adenylate or adenylic acid. Similarly, for guanine, it will be guanine is a base, guanosine is a nucleoside, and guanylate or guanylic acid is the corresponding nucleotide. For uracil, uracil is the name of the base, uridine is a nucleoside, and uridylic acid or uridylate is the nucleotide. Cytidine, cytosine is a base, cytidine is a nucleoside, and cytidylic acid is a nucleotide. Thymine, thymidine, thymidylate. Okay, so now you know the different names. When to use what? When we are saying nucleotide, we must end with dilate. Guanylate, adenylate, cytidylate, uridylate. Right? It's acid, phosphoric acids. Next. Purine biosynthesis has got two major pathways. One is de novo synthesis and other is salvage pathway. De novo means the new, means forming a new molecule starting from the scratch. Salvage pathway means reutilizing already manufactured something is there, and we are, instead of throwing it out, we are again reusing it, almost like recycling. It's called salvaging or salvage pathway. Right? Next. Now, as I told you, the first point to be remembered in de novo synthesis is the molecules which contribute the molecules in purine. What molecules contribute to the other sources of different molecules of purine, you have to remember. This is very, very important. 1, 2, 3, 4, 5, 6, 7, 8, 9, like that. And then which carbon, which nitrogen is donated by which compound, you have to remember right hand side that table. To remember the table is very easy. See, 1, 2, 3, 4, 6. 2 and 8 will be given by formate. 1 is given by aspartate. 3 and 9 given by glutamine. 4, 5 and 7 given by glycine, means 4, 5, 7. These 3 given by glycine, and this one 8 and C2, this one and this one, they are given by formate. Which formate? N10 formyl THF, right? Folic acid. I told you folic acid is required for synthesis of nucleic acid, right? C6, the topmost carbon, is given by bicarbonate. So this is very, very important. Very often this question is asked. Very often. Show the different sources of different molecules or different atoms of the purine and pyrimidines pyrimidine bases or pyrimidine nucleotide. Okay. Next. Now we will start the de novo pathway. The pathway contains 15 steps. Okay? 15 steps. The good news is, this is very rarely asked. The bad news is a part of it is asked. Correct? So which part will be asked, that I will tell later. Fine. So we will start with the pathway, the first reaction. Remember, all synthetic reaction mostly takes place in cytosol. So it also takes place in cytosol. But in this particular step, this particular pathway, you will find many steps where ATP or energy rich substances are required. So remember this point, that any enzyme whichever is involved in oxidation of this energy rich compound are always membrane bound. So the enzymes which utilizes ATP, GTP, CTP, whatever it is, as for production of energy, then it must be membrane bound. The reaction starts with alpha D ribose 5-phosphate. Alpha D ribose 5-phosphate. Can anyone tell me what is the source of this ribose 5-phosphate? Which pathway? Anyone? Which pathway? Bolo? HMP shunt pathway. HMP shunt pathway is the source of ribose 5-phosphate. Anyway, so the first product is transfer of 2-phosphate and to form phosphoribosyl pyrophosphate, PRPP, phosphoribosyl pyrophosphate. Just write down the names. Do not try to draw the structure. Just write down the names. Ribose 5-phosphate, then PRPP, phosphoribosyl pyrophosphate. Enzyme kinase, phosphokinase. Okay. 1 molecule of ATP is converted to AMP. The 2-pyrophosphate is transferred to the 1 position of the ribose sugar. Next, the next step, PRPP is converted to phosphoribosylamine. Phosphoribosylamine. Amine is added from glutamine. Now, this kind of reaction always require glutamine plus water, means glutamate plus PPI. If this compound was not having pyrophosphate, then this particular reaction would have required one extra ATP that we will see in some other reaction. But because ATP is already added in the previous step, we do not need an ATP to transfer the amine group. We do not need ATP to transfer the amine group. So this is amidophosphoribosyl transferase. Amidophosphoribosyl transferase. Next. Now, the ribose is then added with one glycine. Ribosylamine plus glycine. This is glycine. This is glycine attached with one amino group, CONH called amide. So this is glycinamide and this is ribotide. Name is glycinamide ribotide. You can remember this name. Very easy and lucrative name. Whichever you want, you can remember. It's up to you. This is glycinamide ribotide. Next. Most of the enzymes are synthetase. Then, wherever formyl group is there, the enzyme is formyl transferase or transformylase. Correct? This one, glycinamide ribotide, this is glycine. To this one, formate group is added. Formyl transferase. So N10 formyl THF to THF and this formate group is added here. So what it becomes? Formyl glycinamide, ribotide. Ribotide means ribose plus sugar, phosphate, ribotide, which is added with glycine, which is added with amine. So it becomes glycinamide. To that glycine again we add one formate. So formyl glycinamide ribotide, or FGAR. Next. Long way to go, only step 4, 5. Fine. The next step, see, this is an example, as I told, in the first addition of glutamine amine, there was no ATP, but here ATP is required. Why? Because the starting material has no high energy bond. So ATP plus glutamine plus water forms glutamate plus ADP plus PI, and it forms formyl. Can you read? Just read the name. Let me see you can read or not. Formyl glycinamidine. Formyl glycinamidine means one more amino group is added. Formyl glycinamidine ribotide. The previously it was formyl glycinamide ribotide. Now we have added one amino group, so it becomes amine of amine. So formyl glycinamidine ribotide. Next. Next step, it doesn't do anything. It just cyclizes the whole molecule. It forms a cyclic compound. The first cycle is formed in this step. Enzyme is very simple. All the enzymes are mostly synthetase, if not transferase. Okay, mostly synthetase. See, ATP is required, and this is joined. When this is joined, this excess amino group is attached to what do you call this ring? Ring structure, this ring? Imidazole. So one imidazole attached to one amino group. What do you call? Amino imidazole. 5 amino imidazole, 5 amino imidazole, and that is attached with ribose sugar. So ribotide. So AIR means 5 amino imidazole ribotide. The name is big, short name is easier to remember. AIR, 5 amino imidazole ribotide. Next. AIR. Now the things are becoming more and more complicated. Names will become much bigger. Okay. Just add one C in front of A. That is carboxy. What is that? The 6th carbon is added here by bicarbonate. AIR carboxylase, 6 carbon. See, this is added. So carboxy amino imidazole ribotide. Carboxy amino imidazole ribotide. Right? CAIR. Next. In the next step, one aspartate is added, whole aspartate. Whole aspartate is added. This is one of the 2 reactions in the whole pathway, same reaction, where aspartate will be added, and in the next step, one fumarate will be removed. So aspartate is added through what? Through its amino group, where? To that carboxylate group. So now this whole thing is attached to what? This amino group, this nitrogen. And if you say this one, then it becomes 5 amino imidazole, this one 4. 4 is attached with one N. Carboxamide, one of the N hydrogen is replaced by this. What is what? If you don't look at this nitrogen, amino group of aspartate, what is this compound? This is succinate. So, as if it is a succinate group attached to the amide, nitrogen of the amide of the carboxamide, and that's why it is written 4-aminoimidazole, 5-aminoimidazole, 4N-succinylo carboxamide ribotide. Right? This is how it is named. You know how it is named, you know the structure, that will be easier to remember, even anyway, to understand the things. Okay. So, SAICAR, 5-aminoimidazole, 4 within bracket N-succinylo carboxamide ribotide. Next. Adenylo succinate lyase. I told you there are two such reactions. So, first reaction aspartate will be added, the second reaction fumarate will be removed. If fumarate is removed, what is left with? The amino group of the aspartate. So, who are donating a nitrogen group? Aspartate is donating nitrogen group, glutamine is donating nitrogen group, and glycine is donating the whole molecule. Right. Now, this becomes 5-amino 4-carboxamide ribotide. The succinylo is not there, fumarate is removed. Now you can say, like, sir, you are removing fumarate and previously name was succinate. What is the difference between succinic acid and fumaric acid? I don't want to complicate, forget it. Anyway, next. The last but one step, another N10 formyl THF is required, which donate is formate group here, this black one, to form ribose 5-phosphate attached with one imidazole ring, which has got one formamino, formamino imidazole 4-carboxamide ribotide. Formamino imidazole 4-carboxamide ribotide. This whole thing. Next, last step, it is again cyclized to form inosine monophosphate, the basic compound, the starting compound for synthesis of other purines. Right? Other purines. Inosine monophosphate. IMP cyclohydrolase. Now, up to this, you are not required to remember. Very rarely they ask. If this year they ask, then that is your most bad luck. Okay? 99% chance you can, it will never be asked. Each reaction of one side is supplemented by or complemented by, the product of the reaction from the other side. So, from IMP, if you take left hand side this way, the first is IMP dehydrogenase. It forms XMP, means xanthosine monophosphate. Xanthosine monophosphate. Then one amine group is added, it becomes guanosine monophosphate. Enzyme is transaminidase. Transaminidase. Sorry, amidinase. Transaminidase. Transamidinase. Amide group transfer. Okay. Then this side you go, again this aspartic acid is added and fumaric acid is removed. So, adenyl succinate synthase and adenyl succinate lyase. One is adenyl succinate synthase, another is adenyl succinate lyase. Left hand side IMP dehydrogenase, transaminidase. Right hand side adenyl succinate synthase, adenyl succinate lyase. Add one aspartic acid, remove one fumarate. You get AMP. In between you get adenylosuccinate and then you get AMP. Here you get one more XMP, GMP. IMP, XMP, GMP. Okay. So this one is very often asked. Next. Regulation. Next. Remember this. The whole pathway function or is regulated by feedback inhibition. By feedback inhibition and product activation. Product activation. See, this PRPP stimulates the synthesis of PRPP itself. It is not shown here. It's called product activation. Rest all are feedback inhibition. AMP inhibits the first reaction, ADP inhibits the first reaction, GMP inhibits the first as well as second reaction. What is this reaction? Name? Enzyme? PRPP synthase, correct? And this is transaminase. GDP and GMP both inhibit this one. AMP and ADP both inhibit this one. GMP, on addition to that, it also inhibits the second reaction. That's all. Is there any other reaction? Yes. How they are regulated? I told you this is not shown here. PRPP stimulates the synthesis of itself. The ATP, mostly I have shown you the negative regulation. The positive regulation is positive regulation is PRPP, product activation, and ATP and GTP. If you can recall, the synthesis of ATP requires GTP. Which step? Adenylosuccinate synthase, it requires aspartate plus GTP, right? And then the synthesis of GTP or GMP, it requires ATP, where glutamine, the very first reaction, glutamine plus, last reaction, glutamine plus ATP. So in both cases, for synthesis of GMP, ATP is required. For synthesis of AMP, GTP is required. So if the cell produces more GTP somehow, so the stimulation points are 3 points. ATP stimulates for formation of GMP, GTP stimulates formation of AMP, as well as PRPP stimulates synthesis of itself. It's called product activation. Next. Okay. Now we come to the salvage pathway, then we stop for today's class. Salvage pathway is like, suppose we are consuming a lot of DNA and RNA every day, in the form of vegetables, in the form of fruits, meat, egg, whatever it is. So those DNA and RNA are absorbed in the form of nucleic acids, right? Nucleotides. Those nucleotides, when they reach the liver or the other tissues, again, during synthesis of DNA, during replication process, in transcription process, in translation process, sometimes some DNA and RNA are destroyed prematurely. So that is also produce a lot of debris inside the cell. These are broken down to the individual nucleotides and individual nucleotide pools are made. When required, we can synthesize these nucleic acids from them. This is called salvage pathway. Like we have adenine, so we can add one PRPP, we can form one AMP. Very easy. Previously what we did, we synthesized, we took ribose 5-phosphate and then we synthesized the whole purine ring on it. Correct? Here what we are taking, we are taking the already synthesized adenine and just adding it to the PRPP. So we get AMP plus PPI. Similarly, for hypoxanthine and guanine, we get IMP and GMP. Once we get IMP, we can synthesize IMP from, we can synthesize AMP or GMP from IMP. Right? It can be used as a raw material for synthesis of AMP and GMP. Moreover, GMP can directly be synthesized if we have guanine in the cellular pool. Right? These two reactions are catalyzed by APRTase and AGPRTase. Adenine phosphoribosyl transferase and hypoxanthine guanine phosphoribosyl transferase. These two enzymes are very, very important. If these two enzymes are not there, then cells become, cells accumulate a lot of nucleic acid debris. They cannot utilize it. Nucleic acid debris সেলের ভিতরে জমা হয় আবর্জনা স্তূপ মতন ঠিক আছে সুতরাং এই পার্টিকুলার দিস টু এনজাইমস আর ভেরি ভেরি ইম্পর্টেন্ট স্যালভেজ পাথওয়ে ইম্পর্টেন্ট বটে কিন্তু এইগুলি আরো বেশি ইম্পর্টেন্ট কারণ এগুলি না থাকলে স্যালভেজ পাথওয়ে না থাকলে ডিএনএ তৈরি হবে না এগুলি না থাকলে absence of these or hyperactivity of these will create clinical condition. Absence of this will create Leishman syndrome. Hyperactivity will create gouty arthritis. In both the sides, there will be problem. APRTase and AGPRTase. Adenosine, adenine, phosphoribosyl transferase, and guanine, hypoxanthine guanine. Enzyme is same, but their starting material are different. Name of the enzyme is same, hypoxanthine guanine phosphoribosyl transferase. It can transfer either hypoxanthine or guanine to phosphoribosyl transferase. Clear? Any question? Almost all cells perform this particular pathway. So if they suffer from deficiency of this enzyme, will lead to a condition called Leishman syndrome. Okay, na? Leishman syndrome. LES SH, Leishman, NYHAN. LESCH, NYHAN, Leishman syndrome.